get this party started. So just to kick us off a little bit, a few housekeeping rules before we start the session. First of all, please make sure your phones are on silent. Feel free to take a moment to double check them. I know some people think they're on silent, but they're not. Photography of ABRF meeting sessions is permitted, subject to speakers' request not to capture pre-publication information. Recordings of the program sessions will be available on the meeting website following the annual meeting. ABRF's social media policy is that everything can be shared. If a speaker asks that it not be shared, please respect the speaker and do not post anything on social media. If you are going to share anything on social media, please use the hashtag ABRF2024. Unless noted by the speaker, questions are to be held until the end of the session, including questions for the author of the lightning talk. So I hope your memories are good because you got to wait till the end for the lightning talk. Attendees may ask questions at microphones in the room. This is because we are recording the session, so we want to make sure that we can hear your voices. Download the ABRF meeting app to view the full schedule, access lightning talks, abstracts, connect with other attendees and sponsors. We also encourage you to play the ABRF game and create your personalized meeting schedule. You'll find extra content, including hours of the exhibit hall and tech showcases to thank our sponsors. And session and lightning talks can receive also a 1 to 5 star rating in the app, in addition to attendees who will receive an overall meeting evaluation survey following the meeting to help improve the ABRF meeting experience. Any questions on the rules? First, I'm going to introduce our vendor talk, who, without their support, will be able to present content and organize conferences like ABRF. So I'm going to introduce Scale Balance's Christina to come up and present.
All right. Hi, everyone. So, like I was introduced, I'm Christina. I'm the local representative for Scale Bio. And so, in case you aren't so familiar with us, Scale Bio is a single-cell sequencing solution company that is based out of San Diego. I'm also so excited to be part of this particular session So Scale has really managed to take advantage of all of our collaborations that have been with both academic and then also commercial partners, and we're going to talk about that a little bit more in the coming slides. So being part of this section of facilitating these cross-functional studies is really very useful. So in case your background is not in single-cell sequencing, just a little bit of history. This is a technique that has been around for about 10 years and has actually been truly pivotal in actually advancing both our understanding of human health and disease, as shown here. Oh. Okay, advanced in the slides, but not actually over here. Okay, so.
Tech man.
I have already broken it. I'm so sorry, guys.
Mr. Tech Man in the corner, back there. Hi. Slides aren't moving. We're like using arrows and they're not changing.
I might go over 2 minutes past at this point. I'm so sorry, guys.
We're in the penalty box right now.
I know. Oh. Oh, there we go.
Thanks, Dylan.
All right, fantastic. Thank you, guys. Saving my butt. And so, like I mentioned before, single-cell sequencing has been truly pivotal in actually understanding human health and disease, but this is also a species-agnostic technique. So it has moved forward our understanding of molecular biology as a whole. We're back, okay. So one of the things, though, that we have noticed about the single-cell sequencing publications that have already come out is the fact that they are somewhat limited in either the scale or the actual longevity studies. And so what we at Scale are actually able to do is address both of those issues. If you are interested in actually completely looking at the entire tumor heterogeneity or the tissue that you're looking at, you would need more than just a standard 10,000 cells. And we actually offer a solution that allows you to go up to 500,000. If you are interested in doing a longitudinal study, we have a solution that actually allows you to go through and do a methanol fixation. So you can store these samples and then run them all at the same time to eliminate batch effects. We also do this while focusing on cost compression. And so instead of having a capital equipment that you have to purchase and also the increased cost that comes with these solutions, you actually can go through and use ours and keep those costs for this increased number of cells, but also keep the cost down. And, okay, there we go. And so how we are actually able to identify these limitations in single-cell sequencing solutions that have been previously offered is we actually are founded off the ideas of experts. And so if you've been in the proteomics field previously, you've likely come across Gary. If you've come across bulk NGS or done bioinformatics, you've come across Jay Shinduri or Cole Trapnell. And finally, if you worked with Illumina sequencing in the past, you likely have come across our CSO, Frank, who has actually been involved in Illumina for over 20 years as their Director of Advanced R&D Development. And so the idea that we have that allows you to actually get this increased cell throughput while keeping costs compressed is the idea of using the cell itself as the partition or the compartment, moving away from the limitations that either come with one cell per well or the actual droplet-based sequencing that is based on Poisson distribution. And so what we have done is allowed you to actually take highly parallelized indexes and add them onto your analyte of interest. And this allows you to take many cells in the individual well to start, but get an individual path through as you add more of these nucleotides on. So we have 3 different products that we actually offer at scale that are based off of this platform of no capital equipment and also this higher throughput from you do this at your lab bench. And so starting from left to right, so we have our single-cell RNA flagship kit, which allows you to actually get up to 500,000 cells. We have our single-cell CRISPR kit, which allows you to get, on top of the actual 500,000 cell transcriptome, also identification of the specific guide RNA that was developed or delivered to this entire cell. And so this is good if you're interested in guide RNA screens. And then finally, we have our first on-the-market single-cell sequencing solution for methylation. And so since we are switching analytes of interest, we're keeping this to 16,000 cell throughput, but it allows you to actually go through and identify in a differentially expressed way the hyper and hypomethylation areas of your cells. We also, though I'm not going to speak about this further, will be offering in the future a solution called Scaleplex, which is a hashing solution allowing you actually to take these very low input samples, combine them together after they have been hashed, and actually treat them as one biological sample. So again, this entire session is focusing on actually having this cross-collaboration, and we've done that with these commercial partners from start to finish in our workflow. So we've leveraged our collaborations from the start to finish, so Miltenyi for actually isolating your cells or nuclei, all the way to base pair to actually do the downstream data analysis. In a couple of slides, I'm going to go a little bit further into talking about our collaboration with SPT LabTech, which allows you to automate our solution. So 3 kits. First one going to be the single-cell RNA sequencing kit, and there are 4 things that you as people who are working with a core facility would be interested in. The first is the fact that this is a very flexible kit. So you actually have the flexibility coming in with the fact that you're going to go ahead and fix these. You can store them for up to a year. This is a plate-based format, so you actually have flexibility in the sample number that you put in. You can put in 1 sample, get 500,000 cells output. You can actually put in 96 samples and get about 5,000 cell output. It's up to what your experiment demands. This is also, the second thing, going to be a very streamlined workflow. It's under 2 days, it can be automated, and it actually has up to a 50% cell recovery with under a 4% multiplet rate. And so this is going to get you very good quality data in a very short time period. And so again, the third thing, this is species agnostic. So everyone starts in human and mouse, but you don't always stay there. And so this, since it is a poly-A-based kit, is going to be able to support these studies outside of that. Finally, the last thing that I've mentioned, and that is really important for cores, is the fact that this is going to actually not require capital equipment, and it keeps the costs compressed. So you're actually able to get more cells with the same amount or less money. And okay, so in the previous slide, I showed you the UMAT that was actually of almost 700,000 PBMCs. But again, people don't usually stay there. So we wanted to show you that for our platform, you can actually go through and using 4 very different types of tissue, liver, lung, and kidney, and brain. You can actually add them all together onto the same plate and get over 400,000 cells out. So this allows you to do a mini atlas and truly characterize what is going on for these different types of cells. And so we also saw this beautiful characterization, but when you looked at a little bit further in the brain, which is a bit of a more difficult sample to actually go through and do single-cell sequencing because of lipids, we got enough sensitivity that you actually could see all of these different types of neurons and glia, but also some of the smaller occurring populations in there. And so I've shown that this one actually has worked with different types of cells, but it also will work well with an automation platform. And so again, SPT LabTech is we've actually been collaborating to do this with. And what we found is that there is no batch effect between a highly skilled technician preparing these libraries just at the bench, or if you wanted to automate it onto their Firefly machine. And so this is something that you could trust the data that comes out that would be consistent each time that you use this. Transitioning away from the RNA, so this has all of the advantages of using the single-cell RNA kit, but we've added on a reverse transcriptase primer into this kit, allowing you to specifically characterize what guide RNA is delivered to each cell. So if you have people doing these high CRISPR or high throughput CRISPR screens, this is a good solution. And so again, we've done this in cell lines where we get to identify over 90% of the actual guide RNAs delivered. However, we've also done this in organoids that allows you to actually go through and characterize up to 71% of the actual guide RNAs that are in there. We've also found that this delivery of guide RNAs does create differences in the population, as you can see in cluster 6. All right, almost done. And so the final thing that I do want to speak to you guys about is the fact that we do have a first-on-the-market solution for our single-cell methylation sequencing. And so this kit allows you to characterize 300 to 500,000 CPG sites for your actual, whatever your input is. And similar to the other ones, this is a high throughput kit. It's up to 16,000 of the cells that you're actually able to characterize our methylation sites for. We have found, so again, first on the market, so it's a complete solution. We've found that this is compatible with target enrichment. So if you are interested in a particular area of methylation, you can use the twist or another type of enrichment protocol with this. And again, this is all capital equipment free. You can do this at the bench. What I've shown here is going to be an internal study that we did looking at an IDH mutant glioma sample, and so human sample that was actually tissue indissociated. For this, what we found is that there were very distinct clustering based on the hypermethylation areas of this data, but you also found that, so you had very strong differences in the hypermethylation, but you also had differences in the hypomethylation, allowing you to delineate this population of cells as neurons and this population as the cancer stem cells. And you can see this shown a little bit in more depth here. And so I'm just at the end of my time. If you have questions, please come to booth 211. We'd love to talk with you guys further about this. So thank you all.
And thank you, Christina and Scale Biosciences, for your generous support of this session. We appreciate it. So next I'm going to have Sam from Becton Dickinson come up, and he is going to be giving a flash talk, which I'm going to encourage each of you to rate in the app on the stars. It helps the education committee evaluate these presentations. I can't find the cursor on this thing now.
I can do that.
Thanks. Did you find it?
No.
Nope, you lost it too. Dylan, we need your help again. I'm not crazy.
It's on the main screen, so you may want to swipe left or right to get back to your screen.
Thanks. Oh, found it. Oh my gosh. I think the presentation setup is just funky.
Yeah, I don't know how it did. Wasn't
Okay.
it this one?
Yes.
I'm glad I'm not the only one.
This one. Okay. In our next session, we're going to teach you how to use PowerPoint. Okay, there you go.
Alrighty, thank you. My name is Sam. I'm a scientist with BD Biosciences, and today I'm excited to share some application data with our new non-fixative sample preservation buffer. So single-cell multiomic workflows are long and often require fresh samples. This can be constraining in your study design and even limit certain cell inputs. To address this need, we've come out with BD OmicsGuard sample preservation buffer. Here, we've applied it with human cells and mouse bulk spleen tissue. Preservation for up to 72 hours, and we've sequenced with WTA, whole transcriptome assay, and CiteSeq. This is an easy reagent to use. You simply put your sample in the buffer, stick it in the 4C, high cell retention, no extra washes. And we've captured the single cell with our BD Rhapsody HT Express single cell capture system. So this is correlation data of non-preserved versus preserved samples at 24, 48, and 72 hours, and what we see is tight correlation across these time points. We see the same thing with site-seq data. This is a 30-plex, as well as flow cytometry. So we show good preservation of mRNA and proteins for up to 72 hours. Next, we look at cell type distribution. There's no bias in cell type preservation. And let's take a closer look at neutrophils. These are known finicky cells in single cell analysis. Gated on the LY6G cluster, we see no difference between the fresh versus the 72-hour preserved sample. Finally, I'm excited to show some preservation data in the VDJ assay, full length. We see high pairing efficiency between the preserved and the fresh sample. And in secondary analysis, we see the preservation of the top CDR3 clonotype in the CD8 cluster. So what this means is that this reagent preserves even highly specific TCR and BCR transcripts. So in summary, I wanted to introduce a mild PFA-free reagent that maintains mRNA and surface protein integrity for up to 72 hours. This is an easy buffer to use, and it's compatible with multi-omic studies as well as flow cytometry. Hopefully it can help you guys expand study design in these types of studies. Please feel free to reach out and scan the QR code for more information. Do we have time for questions?
At the end.
Okay. Thank you.
Okay, and if you have questions for Sam, we can hold them till the end, so scribble them down if you want to remember them. All right. Finding the other PowerPoint. Got it. All right, so the session that Kelly and I are going to present to you is really a current work in process. I'm going to start out by just kind of sharing a bit about the profile of Cincinnati Children's, the number of shared facilities we have, and some of the challenges and feedback that we've been getting from our investigators and how we can improve customer service and optimization of data file delivery to them, especially when samples have to navigate across a number of different core facilities for one entire value stream workflow. And mostly a lot of this does impact areas like single cell. So I approached Kelly one day and said, hey, I have an idea. Why don't we try to work on optimizing something that doesn't exist yet, but try to make this process a little easier for investigators? And I actually presented this idea, she doesn't know this, to like 3 or 4 other core facilities. They're in the same value stream. And she was the only one that kind of took the hook and was really hands-on and did a lot of beta testing for me, and at some point was moving a little quicker than me. So I appreciate a lot of effort. So I'm going to try to not spend so much time on my section and give her more time to talk more about the details and show you the more deeper aspect of the case study that we're going to share with you. So I am not having good luck today. Advance. Nope. Okay, how'd you move the slide, Sam? I'm not getting used to this computer at all. I don't know what just happened. It's on. This is one of the craziest technical sessions. That's what I have. It's not clicking. It's really weird. There we go. All right, so at Cincinnati Children's, we have about 28 shared facilities. We call them shared facilities. That exist in 12 different divisions. There's about 500 employees that work across these shared facilities. Between them all, there's over 150 different instruments and 1,000 different services. So it's a lot of resources that we provide to investigators. And we also have all of these facilities operating on one billing system. So we've absolutely centralized these processes for orders, reservations, and management of those processes, and we average about a 90% cost recovery across all of our shared facilities. These are a list of all of the facilities that exist within Cincinnati Children's, but really what I'm trying to drive here is that we really span across a large area of disciplinary work: basic sciences, imaging, clinical, translational, and recently we added qualitative services, so survey design and survey analysis and interview things. Our biggest challenge across all these shared facilities is managing the different silos and structures in which they exist. So there's 28 of them, 12 divisions, 12 different ways of operating, 28 ideas of running your own shared facility. So how do we move across these silo structures and create a system that allows for it, but also allows for them to preserve their own autonomy and their own identity at the same time? So we wanted to kind of remove the top-down hierarchical structure idea and really just think, we're in this together. We're collaborators. We are scientists at heart. We don't really move in a top-down structure, so we need to identify all the various points that we intersect. And so this is really just a quick, we wanted to become more interdependent versus dependent. And a lot of us work within academic organizations, so we know how much these dependent structures can operate sometimes, and our blinders can get turned on, but how do we kind of carve out that time to become more interdependent across not just how we talk to each other in the hallways or interact on the various scientific things that we're working on, but how we move things like samples through various different core facilities. That was our biggest challenge. We also wanted employees to have opportunities and tools to work productively across these silos, and just bringing things across different divisions and different facilities was really a big challenge for us to try to figure out, because we all have opinions, we all have different ideas and ways of doing things, so how do we kind of unify some of those processes? We really had to work within the culture and structure and identify those functions that we could all identify with and want to work within, and we also wanted to make sure that we had a lot of stakeholder input on what needs to change in order for us to adapt and meet the needs of what researchers were looking for. And when I'm saying this, I mean a lot of the problems that we were having across these core facilities, especially in the genomics area, is a lot of them are trying to pull together information from different sources. So we've got metadata, we've got the actual data off the instruments, we've got the shared facilities that still need to facilitate their billing and know what's coming in the door, and they also need to know how to pass it on to the next door. So bringing together all those different interfaces was really a challenge for investigators, and a lot of investigators were getting really frustrated because they're going to 10 different people getting 10 different answers, and we really needed to find a way to kind of identify a solution to bring all of this together. So the problem was, in this case, we have 4 different core facilities that facilitate one sample workflow. Sometimes they'll start in the flow core where they do their cell sorting, and then they'll move to our single cell genome facility. And unlike some other institutions, our sequencing facility is different from our single cell facility. So now the single cells need to go to the sequencing facility. And then once they're sequenced, they need to go to our biostatistics group for the analysis. So we've got 4 core facilities. When an investigator just wants them to go through one process, they don't care who it has to go through, they just want their data. So how do we bring all these different facets together? So we wanted to include the PI transparency and the status in which those samples are moving through several facilities. We all have great access to these centralized billing systems, but one of the things is that sometimes a PI doesn't know where the sample is in the process, especially when there's 4 different facilities involved. So how do we increase the transparency so a PI can even just log into a system and see where it's located instead of shooting off 4 different emails and trying to identify where it was and then counting on someone to respond in a timely fashion? We also wanted to maintain the identity and business responsibility of each shared facility. So every shared facility has their own services, their own fees, their own needs for what they need to facilitate. So we wanted to actually still preserve that autonomy, but still build a way for them to connect these processes together for that investigator. And then finally, we wanted to make sure we were tracking the files associated with the actual sample or samples that were being submitted. And that is something that can also get very muddy because one facility might handle a different file type and use a different nomenclature, and then another facility will get the samples and they'll use a different type of nomenclature. So how do we really bring together that nomenclature issue to help the investigator know what files are what? And then we also wanted to make sure that our end-to-end workflow, which involved the 4 different shared facilities and 4 different divisions, was somewhat unified. We didn't want the PI to be worried. We didn't want the shared facilities to worry. So how do we alleviate a lot of this stress and burden from all of those different points of contact that happen? So the goal was to create kind of like an Amazon experience. We all know Amazon. My husband hates me for it. So how can a PI just go in and place the initial order? Doesn't matter which facility they start from in any of those 4 that I identified. How can they just submit the order, be able to give the initial information to the first share facility, and then that share facility can just take it on and pass it on? How do they pay for their services? Well, honestly, they don't really care about revenue accounts or who pays or who's collecting the money. They just want to make sure that the right budget number is charged, and they just, scientists don't want to deal with finances. And then finally, how can they track the progress of the sample as it's moving through the system? This was really the most important aspect for a lot of investigators because a lot of things are time sensitive. Hurry up, I got a grant, it's got to do. It was due yesterday, So how do they track more transparency, and then how do they collect and maintain all of that data? The biggest problem was, especially with our biostatistics group, where they'd be like, okay, which share facility did you use? And the PI wouldn't know which ones they actually used. So how do we just kind of mentor them through that process and help navigate that for them? And then manage this across all stakeholders. So the challenge was manual coordination, so steps can be forgotten in each and every share facility. Everybody has their own process. Everybody has their own way of doing things. Challenges in coordinating billing, so sometimes when a sample is moving through 4 different facilities, every facility is trying to get a budget number from a PI. Well, when you're sending out 4 emails instead of 1 at the beginning, you're adding a lot of time to the process. Finally, the PI is confused. I don't know what's going on. I don't know who I'm talking to. I don't know what I need to do. My lab manager just said something. So there's a lot of telephone communication issues going on. And then there's communication gaps because they're person dependent. So we wanted to really think of ways that we can kind of solve for this solution. Do we need to develop a new software system? My administrator hat saying, heck no, I don't want to do that. Is there something that exists within Cincinnati Children's that we can already use to solve for some of these problems? We didn't really have a perfect fit or an idea on what we could use for this. So we just really kind of got our thinking hats together, and I poked a couple of brains. Some of them are sitting in this room on what we could possibly do as far as developing and optimizing these processes, because nothing was really out there that I knew existed. So what we actually identified, and this is not a sales pitch or a marketing pitch, is I actually talked to Stratacore, who is the centralized billing system that we use. I said, hey, I've got an idea. Do you think we could do this? And Kelly's going to share more detail on what it is we did. And Matt, of course, was like, yep, absolutely, we can do that. That's easy. I'm like, Matt, why aren't you advertising this with people? Because this is a problem. We need more of this. So really within 5 minutes, he had some sort of prototype set up with me. I think we were at CTLS talking about this. And this is what we've come up with. So we are moving from this nice long value stream of silos to a value stream where we can increase customer service, transparency, the overall operations of how these samples are moving through the various core facilities, and overall, just in the end, the complete service delivery of what it is we're trying to do, trying to simplify things in a complex world. So the solution was to, we had several stakeholder ideas. We tried and divided many ideas. And then, as I mentioned earlier, this solution actually already existed within our billing system, but we had to exhaust all of our options before I came back to what we already had. So with that, I'm just going to give a nice, warm introduction to someone who's been really putting the dirt into the work, and that's Kelly Rangel. She works in our single-cell genome facility, and she's going to take it over and do a deeper dive on what this process actually looks like, and I'm excited for her to share what she's been working on.
Let's see if I can operate this as well. All right. So as Nicole mentioned, my name is Kelly Rangel. I work in the single-cell genomics facility, and I just need to take a couple of steps back and identify what Stratacore is, what basic Stratacore projects are. As Nicole mentioned, she had tasked me with how do we get multi-facility projects moving in Stratacore. And actually, give me 1 second. Yeah, does this not populate the notes? I just realized that it's not moving through these either. Okay. Anyway, okay, I'll continue on. So we use our, on the right, we have all of our shared facilities that are within StrataCore. And our users can access StrataCore to do order services, book time, and reserve assisted assays for things in our facility, such as the Chromium X single cell workflow. But StrataCore isn't just a user to shared facility interaction. Shared facilities can interact with our users as well, and that is through, we do their favorite thing and we bill for all the services. Of course, Nicole already mentioned that. But shared facilities can also contribute to projects and help to contribute to those projects and monitor the progression of those projects until they are complete. All right. Oh, great. Now I've broken it. Now it's moving on this screen and not the other one. So what did I do? It's moving on this one. And not
I've
this
been
one.
having problems all day. Help. They didn't give session organizers tutorials on this.
Sorry, Dylan.
It's not moving.
It's moving on this one instead of on both screens. New presenter's worst nightmare, right?
So, how many engineers does it take to screw in a light bulb? Anybody? None. It's a hardware problem. My husband's an engineer.
Okay, before you run away.
Bazinga.
Okay, so it's not moving on here, though. Is it supposed to move on here as well? Because I know as soon as I click it.
Yeah, go ahead. Kind of. So all of our financial and billing data and our usage data does go in through StrataCore. We do have some facilities that use an API to capture like time-based billing for people. And so that's another system that we feed into an API. But yes, all of the data that I use for metrics at the institution is all in StrataCore. So it's all in one spot.
I'll just do my best to go from memory what I had.
Yeah. I mean, without that API flexibility that StrataCore afforded us, I mean, it would have been really hard for us to understand what's going on in all the different core facilities. And so one of our ultimatums, if you will, is that if you're going to be a core facility and you want to receive a subsidy from us, your billing data has to be able to flow into StrataCore. And so that did require some conversations, some modification of processes, but eventually we were able to provide that flexibility so people could use another system if they want to and develop the API, and it just pipes right on through StrataCore and it bills there.
Okay, I'll just do my best with this.
There's hope.
Okay, guys, I'll do my best with what I have here. So we'll get into what is a StrataCore project. So our shared facilities can contribute to StrataCore projects in order to help our users understand what samples they've submitted so far, give them sample notes, etc. And we can even go as far as contributing some small QC files for, DNA QCs, RNA QCs, et cetera, that are needed for them. But a StrataCore project is a collection of all of these items that are pertinent to an experimental workflow that's found within a shared facility. All right, so we have all of these items being pumped into a StrataCore project, and it's specific to a particular workflow. So how is a StrataCore project requested in a facility? First, your user needs to identify that they require the workflow and that it's found in your facility. They're going to go into StrataCore, they're going to submit an online form requesting that project be made for them. When they submit that request, it's sent to everybody in that shared facility so that it's created for them, or we'll set it up for them. Once the project is instated, the user can then use that project to place orders, reserve time, etc., on the various instruments that are present in the facility. And once the pipeline's in order and the user is submitting orders or reserving time, we can track the progress of the project until it's complete. So here I have two back-to-back videos that I'm going to talk through. The first video walks through how a user would submit an online form to request a project through our facility, and then how our facility goes about creating the project for them. And then the second video will be how we as a facility can view all of our projects, the active ones that are there, and then I'll go briefly into what the innards of a project can look like and where we can contribute to them. Okay, so I think this one starts up a little slow, but essentially, like I said, the user needs to know where to find, they need to know your facility has the workflow that they are interested in. For us, I'm going to be demoing our single cell Chromium X workflow. So they're going to know that it's in our facility, and they're going to go to the request tab of StrataCore. Here we're going to see the various different project creation request forms that we have for the different workflows that are provided in our facility. When they click on, say, Chromium X, because they're interested in single cell, they're going to be presented with a form. And that form is going to have 3 different fields associated with it. Every project needs a title, a short abstract of some sort, they can make it as long as they want, and a financial account number. So those are the main 3 things that are required. However, down below here, I have added a whole bunch of information that I am trying to acquire, or questions that will provide information from the client so that I can run their experiment without having to ever send them an email. So for Chromium X assays, I'm asking things like, how many samples are you going to send to me? What assays do you want to run? 3', 5', next gem, version types. And, even as far as what read depth do they want from their samples. Once they fill all this information out, they can submit the request. And at this time, everybody in our facility is notified that this request has been submitted. Okay, so we go back to our homepage, and I see that Ramesh has submitted a request. When I open that request up, I can see all of the information that he has input in this particular project request form. So what we do is we, as a facility, go through, double check all their answers. I have some free text that's in there. They can get quite creative with what they put in there. So I need to make sure it makes sense. And if it does, I go ahead and create their project request form. And StrataCore has this wonderful ability where I can send them a notification when I'm doing this process. That's what I did just there. All right, so to keep our facility organized, we rename all of our projects. I let our clients know this, or our users know this, that we're going to be overhauling their project name. And all of our projects are named with our acronym, the particular assay or workflow that they are going to be interested in. It has a numerical identifier with it and some other information. And in parentheses, when I get there, we add essentially a user identifier. So if one lab has multiple projects in our facility, they can look at this identifier and say, oh, yeah, that's SAT B2 project. And they can see that at a glance, especially if they have multiple projects. So after I'm done renaming that project, I will add individual users to the project. So here I'm going to add all of our single-cell genomics staff. They're going to be labeled as project admins, and this means that we can update or modify anything within the project. while Ramesh is going to remain as project leader. So he can request to have certain things edited about the project, but can't make huge changes to it, because the last thing you want is to be hit when you're halfway through the workflow with a whole bunch of changes. So they're restricted to project leader. Lastly, this is my favorite portion of establishing a project, is adding the group members. So adding an entire group to a project, meaning the entire research lab to this, means that when we're in academia, so we have a lot of graduate students go in and out of laboratories, right? When a graduate student starts a project and then they leave, anybody else in that lab can pick that project right up because they already have access to it and move forward with the project as they see fit. Okay, so now we've made it through essentially steps 1, 2, and 3, where we've created the project, and now the user would be placing orders or making reservations on calendars, etc. And now we're going to essentially track the progress of the project over time and contribute to it. And in this video, I'm going to show you the blue bar is specific to admins that are in a shared facility. We have access to this bar only. I don't think I started this. There we go. So eventually I will click on the projects tab, and we're going to review the whole list of projects that we have that are active in our facility. Give it just a second. And then, so this is what all of our active projects look like. What's great is that you can actually sort based on project type or workflow type, all right? So here we had all of the forms that they could request. Essentially, these are the projects that are associated with them. And you can sort based on your workflow or project type. Here, I'll do Chromium X. So every project gets a unique reference number, which is great. So it has a unique identifier associated with it already. However, you can see at a glance what's going on with projects. I would say those first, I didn't do it. The first 3 projects or 4 projects that are up there don't really have much activity. But project number 7 actually has a lot of orders associated with it, and you can even see at a glance a running tally of the costs that they have accrued with the orders they have placed. As we scroll down, am I hitting something? As we scroll down, we can see the different project phases. So these particular sets of projects are in our sent to sequencing phase. So they're almost done. They're being processed. And if I click on this project, we're going to show you the inside details of the project. So we know that Amber saw from bottom to top in the milestone section. Amber was the one who created it. My colleague Xander kindly created the project for Amber, and then I was the one who actually processed many of her samples within the project. And I would add all these updates to them, because once they gave us the cells, they would walk away and they'd have no idea if it failed or was successful. So I'd give them these nice details and reassuring them that everything was successful. If we scroll down, we can see how many orders that they have placed against this project. This section is great for when you have repeat offenders who cancel all the time and actually never bring you a sample. I have only 1 or 2 groups that do that to me. So the last part is the project status changes that you can make. We actually have 4 different phases that we can go through with a particular project. However, I've found that when we have like 60 plus Chromium X projects now, having multiple project phases is a little burdensome to have to update those constantly. So as of right now, we're essentially just using phase 1 and 2, or project start and project finish, essentially. All right. So pros and cons of StrataCore projects. Of course, they increase transparency and customer service. So the clients can see all the orders that they have placed against a project within your facility, and can review all the notes that are there all in one location. What I really loved and already explained is that you can add a whole laboratory group to a project, which allows this project to exchange hands when you have graduate students coming and going from your laboratory. They can pick up where they left off. Some cons is that a project is only as useful as you make it. So the details that you put in are only as useful as you make them. And if you have somebody in your facility that isn't contributing to a project, then the user themselves are not getting that information and may not find the project as useful. I would say that there's limited flexibility when using multiple financial accounts for a particular project. At the beginning, I had showed you that in the project creation request form, a financial account number was required, and that's attached at the project level. So when I go to change financial account numbers at the order level, it dissociates it from the project. Now, if there's an update that I'm unaware of, great, but as of right now, this is one of my cons. Lastly, projects were limited to one shared facility. So how do we handle experimental workflows that require, the use of several shared facilities? And you'll never guess, it's multi-facility projects. So now we have a collection of orders, bookings, all those items that I mentioned before, but they're pertinent to an experimental workflow that's conducted by a predefined group of shared facilities and a subset of their applicable services. So multi-facility projects have all the same pros as just the basic project that's localized within a shared facility, but it achieves a new level of transparency, okay? Again, we can see all of the orders that were placed across all of the shared facilities that are involved in the particular workflow. It allows for monitoring of complex workflows by the user and facilities. So, if our upstream partner, the flow cytometry core, adds notes there, I can review those notes, see what kind of quality samples were coming into our facility, et cetera, and kind of monitor what's going on upstream of my facility versus downstream as well. And it opens up lines of communication between shared facilities. Instead of me having to send a whole bunch of emails to all these facilities asking what's going on with certain sample preps, et cetera, I can just review notes within the StrataCore project. All right, so how is a multi-facility project established? First, you need to identify the shared facilities that are a part of the workflow and then determine what services are going to be contributing to it and what interactions you would really like for the shared facilities to be contributing to that project. Next, you would have to obviously create it or set it up within StrataCore and then allow your users to use that. But as a note, a multi-facility project can only be created by someone like a super admin. So our super admin is Nicole White, and a super admin is essentially the individual who has access to all the core facilities and all of their settings. I'm considered an admin. I only have a specific user or a set of user rights within our facility and access to certain settings. And of course, the user is the individual who's just interacting with the software to place orders. All right, so where are we with actually implementing multi-facility projects? We're definitely in the development stage, as Nicole had alluded to. We've identified a workflow, we've determined the services that would be involved in that, and we're currently setting it up and piloting it in StrataCore right now. So we're starting off small, and we're just going to focus on 2 shared facilities. That's my facility, the single-cell genomics core, and our downstream collaborator, the genomic sequencing facility. The workflow is our single-cell workflow, and the services that are going to be a part of that workflow are strictly the chromium-related assays and all NGS sequencing from the genomic sequencing facility. So of course we wanted to increase transparency, improve user experiences, but we also wanted to try to localize some metadata by using these multi-facility projects, and we wanted to improve some billing processes. So for this slide, I'm gonna walk through a couple of step-by-step slides so that it'll show you how this workflow is occurring. So for the Chromium X workflow, it's going to start in the single-cell genomics facility. And it's going to start by our users going to our booking calendars. And they're going to, because our single-cell assay is an assisted assay, they bring the samples to us and we process them on the Chromium X instrument. They go to our calendars, they reserve time, and they'll use the multi-facility project that has been provided to them, also has a financial account number already linked to it. But when they actually place that session or schedule that session, there is a setting within StrataCore that allows an order to be generated. So we have something to bill against. So when that order is generated, and they had reserved their session, they'll bring their samples during their session, and we'll process all of these samples until they're into libraries. These libraries will sit in a queue until we have enough samples to pool to send to our genomic sequencing facility. Okay? Now, one of the hiccups with I don't want to say hiccups, one of the services that we provide to our users is that we will pool their samples for them and submit them to our downstream facility for the clients. This means that there's no user interaction with the next facility. Okay, so I make this pool of libraries, I walk it down to the genomic sequencing facility, and they go, oh, you're now the user, you're going to get billed for this pool because you submitted it to us and it has your name all over the submission. So what we're trying to do by implementing this particular workflow is get the sample names and the ownership of those libraries into the next facility so that they have a direct path for billing. And we're going to do this by adding a sample manifest to the order to help transfer some of this, what I'm calling metadata. Okay? So to the order, I'm going to add that sample manifest that has sample names, possibly a pool ID so that we can get all the information collected and together in one spot. Here's an example of a sample manifest. All of these details in these columns is something that you can define yourself. You can chat with your downstream facility and go through and designate what is important for them to use. Once the sample manifest has been attached to the order that's currently in my facility, we're going to enter what's called an order phase change. This is what an order looks like. You can see here that it already has, I'm still using Ramesh's project as an example. It has the PI's information on it. It would have been his name if he were actually doing this. I'm in the demo environment, so it's gonna have my name all over it. But it would have his name there, his account information, and when I change the order phase from processing in our facility to being sent to the sequencing facility and I click OK, what we're hoping for is this order phase change will generate another order in our downstream facility. Okay, this is something that we are working with StrataCore to do. It's currently not an option. Their software engineers are working with us to make this happen. So when this order gets generated in the downstream facility, it'll have a copy of the sample manifest with it. So now our downstream facility has, oh, these samples belong to Dr. Ramesh, and it's located in pool blah, blah, blah. And they have more information about library ownership, and now they have an order that they can bill against. So this is kind of a top-down view, and I think I'm in the last couple slides here. Top-down view of what I'm trying to explain. So our clients are going to schedule on our calendars. They're going to use different multi-facility projects that are assigned to them. And on their session date, they're going to bring us the samples. Okay? When they schedule, it will obviously generate an order. And once all of their samples are processed into libraries and they've been identified to fit into a pool to be sent to sequencing, we will modify the specific orders that those libraries are associated with and insert sample manifests. The order phase will be updated at that point. The trigger will be initiated, and then a new order with a copy of that manifest ends up in the downstream facility. There should be another box here. So all that important information has now been transferred into the next facility. I can bill from my own orders for my specific services, and they can also do the same for their NGS services. So what challenges have we identified just with trying to implement this system? One of the things that I've noticed is that individual shared facilities use projects differently. In our facility, we use projects as a collection of orders that are related to samples, and all of those samples will be used for comparative analysis. However, another facility that happens to sit adjacent to us in a lab, a project to them is just one sample that passes through a workflow. They will never have more than one order or one sample associated with that project, and it moves forward with their workflow. Another facility, which is actually the downstream facility, the genomic sequencing facility, they only use projects as a placeholder for a financial account number. Other than billing processes, they don't really interact with StrataCore. So differences in sample intake can also be a challenge, especially when trying to merge workflows into a multi-facility project. Our facility uses StrataCore for all sample intake requests. However, our downstream collaborator, the genomic sequencing facility, they don't. They have a standalone barcoding station. They manage their own database. So trying to merge our workflows together is kind of a hurdle that we're trying to overcome, which is why getting an order into their facility is something that we're trying to achieve. Lastly, accessibility and usefulness of metadata. I had mentioned adding a sample manifest at the order level and not the project level. We're not certain how useful that's going to be, especially for our users to find, especially if they're finding the majority of their information at a project level, where they're seeing all the interactions and notes that shared facility staff are contributing to that project. All right, with that, guys, I'll go ahead and end that. Thank you so much for bearing with me. I appreciate your time. I'd be happy to take any questions.
Any questions for Sam first?
Maybe.
You?
Maybe.
Yes. For Sam?
Oh, for Sam. No, sorry, Sam.
I'm trying to give Sam some love here. He waited all the way till the end for questions. No questions for Sam? Do you want to redo your lightning talk so I can remember what you talked about? I tried. All right. Go ahead.
I've got two. One, Nicole, if you're the one creating a multi-core project, if somebody reaches out for a quote, are you the one reaching out to all the individual facilities to get that quote for the PI?
No.
So how does that happen?
So all of our shared facilities have their own services, how much they cost, and so like Kelly would put together the quote for her part of it, but they actually have a really good relationship with our sequencing facility, so they know what the cost of sequencing is, and they just kind of add that part into their quote for the customer.
So you take the lead on pulling everything together for the client?
For our current workflow, yeah.
Okay, second question. When you're creating a multi-core project, does the downstream facility know the samples are coming? Can they see that project from the get-go?
They'll get an alert. So they'll get like an email notification that there's a new project in their queue, and they should pay attention to it.
Mine might be two parts, depends on the first. Do you serve both internal and external or commercial entities and projects?
We serve internal and external customers, yes.
Are there any nuances to the workflow that kind of help ease that through? I know in particular, agreements across multi-core projects, for instance, are a challenge.
Our internal and external customers both have equal access to StrataCore, so they're all treated the same. The only thing that's different is where we send the bill or how it's charged.
Okay, third question. Is there any visibility difference between how projects are shown between internal and external?
No.
Okay, okay, thank you.
All people treated equal within StrataCore.
So you showed on the sort of homepage that there's just like a listing of facilities. How does a person know where to start? Like, I need this workflow. How are you guys marketing this workflow? Because we're just barely getting into the space of thinking about the fact that there should be a workflow pipeline across cores.
So I'm fairly new with getting the multi-facility projects going. However, I believe that it would show up in every facility. That workflow should, so they can request it from any facility that is a part of that workflow. They can at least
It'll
request
kick
that
it back
project.
to you.
Not specifically to me. So say that they're in the flow core, the beginning of the process. They should be able to request a multi-facility project within that facility.
How do they know that this is a thing? Are you guys, like, what's your PR campaign on this?
What is our PR campaign on this? So basically, when you go into like request a project in StrataCore, if her request entry point is called Chromium X, and say we had to start in the flow core, we would call that same request Chromium X. So because you have to set up the entire value stream workflow through all 4 facilities at one time, and so that regardless of where it starts, it can always kick down to the next facility. It just can't kick backwards. So you just have to make sure the workflow is set up so it goes sequentially in order.
Yeah, I get the StrataCore side of it. I'm asking you, how are you gonna, do people just know? Because like when I look at the facilities that you have, I would just go to the DNA core, and then they'd be like, no, you need to go back to, you see what I'm saying? Like, how does the message gonna look to your research community? We're offering this pipeline, and this is how it's gonna work.
That's a good question. So I would say I would put that information in their project creation request form.
Okay,
And I
so it'd
would-
be like frontward facing there.
Yeah, it's going to be
Okay.
right there. They're gonna have to read through that. I'm probably gonna make them agree
Yeah.
to it and that they've read it, and they should know how that pipeline works right from the get-go.
Okay, thank you.
Actually, I think I have something kind of tied into that a little bit. We always do consults with any new customers especially, and sometimes we recognize that they're going to have, multiple cores tied into that, or, multiple people at the table to talk about bioinformatics and other things, that kind of thing. Does that accommodate such a thing? In other words, can you sign up for a consult or something like that, so that they say, we want to have a project that has 10 different cores, that kind of a thing, and then you can pull all those cores in together, or like, how does that work? Or do you know yet how that works?
I'm not certain I know yet.
So we could set it up that way, but we currently don't have it set up that way.
Fair.
There is a lot of flexibility in how you design your workflow.
Yeah, fair
It really just depends on like the culture of your researchers. What do they want? What do they need? How do they function? How they operate best? Like our structure and function works well for us, but it may not work well for everyone too.
Right.
So it's just, that's what I appreciate some of Bosma's processes. They're so agile.
Right,
You can
right.
change them to make them however you want them to work.
Right. And so, I guess then you possibly could do it through there. Like, in other words, just have a link or a click that says, yep, we just want to consult, or something like that.
Absolutely.
Yeah. Cool. So, thank you.
Matt DeVries, City of Hope. This is maybe more of a rant or a cool story, bro, but I guess professionally we can couch it as a use case to justify why we needed to solve this problem. Was the workflow I inherited in my position was the upstream core, the downstream core, they would just take it on themselves to place the order as the core in the next core. So and they're like, oh, we see no problem with this, except the upstream or downstream core has been now, in terms of reporting, deprived of a user, deprived of dollars. When you're reporting your utilization as fraction of all usage, suddenly your sequencing core is reporting, our biggest customer is our molecular path core, when that's actually 20 different customers that the other core can't report. And so we're an iLab institution. It actually has a pretty decent collaborating cores feature built in out of the box to deal with this. It's got issues, but it works pretty okay. It was that aspect that gave me the wedge to change that culture, was when you realize you're now hurting the sequencing core because they have 20 fewer customers than they would normally have been reporting had you used it the appropriate way. So anyway, that's my story.
Yeah, and that's a good call out, Matt, too. Is we all have different software systems we use for billing. Some are homegrown, some are from various vendors. But we just use StrataCore because that was what we had available to us. And I'm sure that there's other vendors out there that might be able to work with you to identify a process. But it's up to us, as administrators and people that are being good advocates for improvement within these systems, to go to those vendors and ask for these things to happen. And so we were fortunate enough to have a company that was willing to work with us and deal with my brainiac ideas of, we need this to happen. So advocate.
So I am excited about this talk because at Emory we also use the StrataCore system. So from the client standpoint, it sounds like they would have 2 different orders, one for single cell facility, one for the sequencing facility. So then they have visibility to the milestones that are listed for both, but they're not in the same message. Is that correct? Do you see what I'm saying? Like, so they have to click on one order, and it looks like the, samples have completed at the single cell facility. Now that second order, I mean, clearly if it's gone to the second order, it probably has gone to the sequencing facility, but like,
Right.
it's still not all in one milestone message. So I'm wondering if that's something we can ask Stratacore if there's a way to merge that.
They're right behind you.
So, yeah, that may be something interesting to consider, because one of the key things that you were presenting is, the investigators are having to go different places to find milestones. That's still somewhat going on here, but it's still way better because for us, there's not as many missing pieces to the second group.
Right, and I believe in the multi-facility project, as long as you're clicking on a multi-facility project, all the milestones are ending up in the same location.
Ooh, nice.
Right,
Okay,
so-
I'd love to
so-
see that. That's cool.
Right,
Okay.
so I would be logging into that particular multi-facility project. Oh, sample, blah, 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 was successful emulsion, and then it moved to sequencing, great. If the DNA sequencing core wanted to come in, they would click on that same project because they would have view to that and be able to update milestones as well. And
Got
it's
it.
all localized for the user or the client to see.
Cool. Okay, let me think if I can remember the other question. Project names. So our core facility does project names completely different from the others that we would want this to work with. Is there any way to modify the project name once it gets into our core so that we can kind of keep that right? So that's the other challenge that I see with this. And I don't know that there's a solution necessarily. It may just be that as the Emory or as the whatever, institution, that we have to just come to terms with how are we going to name these so that everyone can work that way.
I think that would be the method that you would have to
Okay.
sit down and kind of make a compromise, because once that project name is made, you don't want to change it.
Yeah.
It's going to be considered something new at that point.
Got it. Okay, that's helpful. Thank you.
So many questions.
Yeah,
guys.
sorry.
No, you're good.
So I have an interesting question. So it seems like the sample name, right, is kind of the primary thread throughout this entire thing. And that's, I guess, sample submitter generated. So I'm at the Jackson Laboratory, where you can imagine we have a lot of investigators that just think of an idea of an experiment, and then they go to like in vivo breeding services that generate the mice, and then those go to research necropsy, and then those go to histology or light microscopy. And each one of those cores kind of cares about something slightly different, like the block ID or the slide ID or the mouse ID, when really the researcher cares about, like, I wanted 3 BRCA mutant mice of this background. And so we're like carrying through all of this sample metadata that like maybe we don't care about. How do you guys like propagate sample metadata throughout these multi-core projects?
That's something that we're going to have to essentially sit down and collaborate with the different facilities that are a part of the workflow and establish up front what type of metadata you want to collect. And then that would, based on how your multi-facility project is set up, that would need to remain constant throughout the entire workflow.
Kind of related to the last few questions, I would submit that maybe the solution is that every interested party should have its own sample name tied to a sample ID. Your core might name things a certain way, the user has a different preferred name, and each core should be able to give it a name that matches their use, and each user should have their use tied to whatever, in the back of their system, it must be a number somewhere.
There is.
Right, and you just allow multiple different names, right?
Yeah, every project that is created in her facility has a number. That number never changes. And so you can always use that through the entire process. Yeah.
Every user should be able to be allowed to, the scientist should be able to call this BRCA whatever. The sequencing core should be able to call it flow cell whatever, part whatever. And that should be a very easy thing to allow them to craft the way they need.
You can even concatenate each.
Can we make sure to use the mic
Makes it
being
hard to answer.
recorded? We're not gonna pick up if we don't use mics.
Thanks, Natasha.
I guess I just have a more broad comment on the idea of metadata. So you're passing through all the metadata. I'm from imaging. So for instance, all the parameters of those collections, you're passing that through. Am I understanding what you're saying about metadata correctly there, or a different type of metadata?
We're not dealing with imaging in this workflow. So one thing that we didn't dive into much of, because this is something that we're working on for the next year, is in conjunction with how samples have to move through a billing system across the various facilities, we have to identify how we're going to move all those files and like instrument metadata so that the investigator has it in one spot at the end of the day. So we are working with our IS team to mirror the StrataCore process and get them to work concurrently with each other, because I don't want to put all my files in StrataCore. They're going to kill me if I do that. That's a lot of data I'm going to hog up. So I want to make sure I find a way to link the passage of that information as it goes through the process, and at the end of the day, that PI has one folder they need to go to for that project and extract all the files they need and go, here you go, statistician, analyze my data.
Nothing more to say other than I applaud that future effort, because from rigor and reproducibility standpoint, that's wonderful.
There's a lot of
Because that
challenges with
just-
it, though.
Absolutely.
And we're
So
working through 'em.
it hardly exists anywhere, right? We're passing that individually if it gets passed. I didn't say it.
It gets really messy, yeah. That's why I'm glad to have her, who's like willing to pull up the sleeves, help me out, and get this done, so.
Right. Yeah. Yay, science. That's all.
Yay, science.
Hey, Nicole. Congratulations. Thank you. Just mentioning at StrataCore, because we've been quoted here. So the project name is something that you can automatize. So like if you have like your project is always like a unique number, the PI name, the users that requested, I don't know, the timestamp. If you have a rule set that you have and you follow in your core for setting up the project names, we can apply that automatically for you. And that can be slightly different for each core as well. So just wanted to mention that. Now in the StrataCore system, you have a unique ID for projects, so we really don't care what it's named. You can rename everything, but clearly you don't want to do that for functional reason of losing your customers or people getting confused about a project being renamed. But that's something you can do. We also have like additional data you can put in, like additional identifiers can be put as secondary values, so that even if you rename things, you can have the original name backed up and reported back to you.
Still talking about those tags, huh? That I don't use. Hi. All right, one more chance. Any questions for Sam? You're gonna ask Sam a question? Thank you. Sam, you wanna grab a mic?
Sam, thank you so much for sharing about the BD buffer. I have to ask, is it only compatible with the BD Rhapsody workflows?
Not